Welcome to another episode from the Veterinary Research Channel. Today, we delve into a fascinating study that explores the detection of a condition known as nephrocalcinosis in cats suffering from chronic kidney disease, CKD. Nephrocalcinosis is characterized by the deposition of calcium phosphate or calcium oxalate crystals within the kidneys. Identifying this condition in cats with chronic kidney disease is of significant clinical interest. While it is known to be prevalent in humans with chronic kidney disease, particularly those on dialysis, information on its detection in our feline companions has been somewhat limited. Historically, microscopic nephrocalcinosis, meaning that it can only be seen with magnification, has been documented in cats with chronic kidney disease in studies examining kidneys after death. However, reports detailing the detection of macroscopic nephrocalcinosis, visible without further magnification, using ultrasonography in cats have been infrequent. A previous study hinted that ultrasonography might underestimate the presence of nephrocalcinosis in cats, although this was based on a limited number of cases without the benefit of comparative tissue analysis. Given anecdotal observations suggesting that nephrocalcinosis might be visualized more often on ultrasound in cats with chronic kidney disease than previous studies indicated, the researchers embarked on a study to compare different methods for identifying this condition. The primary objective of this research was to compare the effectiveness of ultrasonography, microcomputed tomography, MUCT, and histopathology in identifying nephrocalcinosis in cats. To achieve this, the researchers studied 12 kidneys from 7 client-owned cats diagnosed with azotemic chronic kidney disease. Azotemia, in this context, refers to a buildup of waste products in the blood due to impaired kidney function. These cats had undergone renal ultrasonography while they were still alive as part of a larger, ongoing study. Based on the ultrasound findings, the kidneys were categorized into 3 groups: those where nephrocalcinosis was considered present, those where it was suspected, and those where it was deemed absent. When these cats eventually passed away, a post-mortem examination or necropsy was performed. During this examination, the renal tissue was evaluated using microcomputed tomography, a sophisticated imaging technique, to detect macroscopic nephrocalcinosis. This method allowed for the calculation of both the volume and the area of nephrocalcinosis relative to the total kidney tissue. Subsequently, each kidney was processed for microscopic examination. Thin sections of the kidney tissue were stained using 2 different methods: von Kassa staining, which identifies calcium salts, and alizarin red staining, which directly binds to calcium ions. These stained sections were then examined under a microscope, and the area of nephrocalcinosis relative to the kidney tissue was quantified using image analysis software. The relationships between the findings from the macroscopic methods, ultrasonography and microcomputed tomography, and the microscopic method, histopathology, were then statistically analyzed. The results of the study revealed that ultrasonography identified nephrocalcinosis as absent in 3 kidneys, suspected in 3 kidneys, and present in 5 kidneys. Additionally, 1 kidney was found to have a nephrolith, which is a stone within the collecting system of the kidney, alongside nephrocalcinosis, making accurate ultrasound assessment of the latter challenging in that particular case. The microcomputed tomography analysis showed increasing median values for both the volume and the area of macroscopic nephrocalcinosis in the groups where ultrasonography had indicated absent, suspected, or present nephrocalcinosis, respectively. Similarly, the histological analysis using both von Kassa and alizarin red staining also showed a trend of increasing median values for the area of microscopic nephrocalcinosis across the same 3 groups. Importantly, the statistical analysis revealed a strong positive correlation between the macroscopic nephrocalcinosis area, as measured by microcomputed tomography, and the microscopic nephrocalcinosis area as determined by von Kassa staining. This indicates that kidneys with a larger area of macroscopic calcification also tended to have a larger area of microscopic calcification. Furthermore, the study found moderate and excellent positive correlations between the area and volume measurements of macroscopic nephrocalcinosis obtained through microcomputed tomography, and between the 2 different staining techniques used for microscopic nephrocalcinosis. The excellent correlation between von Kassa and alizarin red staining aligns with findings from a previous study. The von Kassa method works by transforming calcium salts into visible black metallic silver, while alizarin red directly stains calcium deposits red. The researchers noted that the specific pH used for the alizarin red staining in this study primarily highlights calcium phosphate and not calcium oxalate. Their findings suggest that nephrocalcinosis in these feline kidneys was predominantly composed of calcium phosphate crystals, a conclusion that aligns with other research in rats, humans, and cats with chronic kidney disease. The kidney with the nephrolith, which was identified as 100% calcium oxalate monohydrate, exhibited exceptionally high values for macroscopic nephrocalcinosis area and volume on microcomputed tomography due to the presence of the stone. However, after the removal of the nephrolith, the extent of nephrocalcinosis in the surrounding tissue as assessed by microcomputed tomography and both histological stains did not appear to be particularly high. This observation led the researchers to suggest that distinct mechanisms might underlie the development of nephrocalcinosis and nephrolithiasis in cats, despite some shared risk factors. Further research is needed to better understand the relationship between calcium phosphate deposition in nephrocalcinosis and calcium oxalate deposition in nephrolithiasis in feline kidneys. The researchers concluded that their study demonstrates a good correlation between ultrasonographically diagnosed nephrocalcinosis and the presence of both macroscopic and microscopic nephrocalcinosis found at necropsy. This suggests that ultrasonography can be a valuable tool for detecting this condition in live cats with chronic kidney disease. Moreover, the study highlights the potential of microcomputed tomography as a sensitive three-dimensional technique for visualizing and quantifying nephrocalcinosis in feline kidneys. The use of microcomputed tomography to evaluate the extent of nephrocalcinosis throughout the entire kidney at necropsy is a novel aspect of this research. The researchers acknowledged several limitations to their study. The relatively small sample size of 11 kidneys across 3 groups necessitates cautious interpretation of the results. Additionally, there was a time gap between the antemortem ultrasonography and the postmortem microcomputed tomography and histopathology, with a median of 167 days between these assessments. It is possible that the extent of nephrocalcinosis could have changed during this period, with some cats potentially developing the condition or experiencing a change in its severity. While repeated ultrasonography in 2 cats shortly after euthanasia showed comparable findings to the earlier scans, further research is needed to definitively confirm the relationship between ultrasound findings and the postmortem analyses at a more immediate time point. Despite these limitations, this study provides valuable insights into the detection of nephrocalcinosis in cats with chronic kidney disease. It reinforces the potential utility of ultrasonography in identifying this condition and introduces microcomputed tomography as a promising advanced imaging technique for detailed evaluation. Further research with larger sample sizes and ideally with more temporally aligned assessments is warranted to build upon these findings and to better understand the clinical significance of nephrocalcinosis in the progression of chronic kidney disease in cats. Thank you for listening to the Veterinary Research Channel. A link for the reference paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.